Hey 12U, how's everything going? So we're almost done cellular respiration, so we're going to be having a quiz. So we're going to have a quiz next week sometime. I'm thinking right now it's probably going to be around Thursday. So we'll put that in. I'll put that in the news feed today that we're probably going to have a quiz on cellular respiration on Thursday. So there's going to be cellular respiration both aerobically and then anaerobically. We'll have our quiz. That's halfway through the unit. Then we do photosynthesis, and then we're going to have our unit test. Okay. Well, I'm going to go over kind of anaerobic respiration today. Make sure that you watch that video from yesterday. This guy does an unbelievable job explaining kind of the role of anaerobic respiration. So I definitely suggest taking a look at that video from yesterday. So I'm just going to shrink this down. I want to keep this on top, and let's go through our video on aerobic respiration. Okay, so it doesn't look like I might appear there, but it doesn't really matter. Okay, so the main thing here is what we're going to be doing is we're going to be processing cellular or glucose without oxygen. And when we do this, we're only going to go through glycolysis. So we're not going to go through pyruvate oxidation or the ETC or the Krebs cycle or anything like that. We're simply going to do glycolysis over and over and over again. And remembering, glycolysis produces a net of 2 ATP. It really produces 4 ATP, but we require 2 ATP to get it started, so it nets 2 ATP. Okay, so just some little background. Many organisms exist in environments that lack oxygen. So areas like, say, our small intestine where bacteria are growing, and they don't have any oxygen, but they still need to have energy. So they can process glucose still anaerobically, and anaerobically means that without oxygen. So during anaerobic respiration, we're going to process glucose without oxygen, and we're really going to do glycolysis. Okay, so there's an example of one. E. coli, it's in our small intestine. Okay, so here's some background on anaerobic respiration. It's also known as fermentation. Okay, and fermentation is really just the process of glucose in the absence of oxygen. So just some background. So many single-celled organisms, like if you're really tiny and you're like just to say a single bacteria, you don't need to have a ton of energy that's produced through the ETC and the Krebs cycle. If you're only one cell, like a small bacteria, you don't need a ton of energy. So doing glycolysis and producing that 2 ATP may be enough for you. Okay. Us being multicellular organisms, okay, we usually need to have more energy, and we can't survive on just doing anaerobic respiration. We need to do aerobic respiration, just because our energy needs are a lot higher. So we need to go through glycolysis and pyruvate oxidation, all the rest of them. But if we were single-celled, we probably would not need to. Okay, so this is the big part here, is this NADH and NAD+. Okay, so NADH is produced in glycolysis. When we go through glycolysis, one of the products is NADH. Okay, and NADH is the loaded carrier. Okay, remembering that when we see NADH, that H represents that it's loaded full of energy. It's not the usable form of energy like ATP, but it's loaded full of energy, and it's about to carry it. And normally, if we had oxygen, it would carry it all the way down to our last stage of oxidative phosphorylation. Right. So here's our product that during glycolysis we're going to make a whole bunch of NAD pluses. Well, we make NAD pluses we make NADHs from NAD pluses. So eventually, when we keep doing glycolysis over and over again, we're going to run out of NAD pluses because all of them are going to be converted to NADHs. Well, when that happens, when we run out of NAD pluses, we're going to stop glycolysis, because as soon as we run out of these, glycolysis can't continue, because glycolysis to continue has got to convert NAD plus to NADH. Well, if I don't have NAD pluses, then I can't convert it to NADH, and glycolysis is going to stop. So this is the whole role of pyruvate in this reaction during glycolysis. Is pyruvate is going to basically take this H from NADH and convert it back to NAD plus. And when pyruvate does that, it turns into lactic acid. Okay, so let me go through what I have here. So however, during glycolysis running and not the ETC or pyruvate oxidation due to lack of oxygen, the cells run out of oxidized NAD+. This is the oxidized version. Remember, loss of an electron is oxidized, and its loss is the electron, so it's oxidized. All are in NADH, reduced form. Gain of electron. Leo's the line says GER, G-E-R. Gain of electron is reduced. So this is the reduced form. So organisms have developed several ways to reoxidize NADH into the form of NAD+, so that we can have glycolysis keep running. Because if we have all of our NAD pluses in NADH mode, glycolysis is going to stop. So this process of reoxidation of NADH by organic molecules is really what's called fermentation. And what's going to do this in this case is going to be pyruvate. Okay, so this is the big thing that we have here is we have NADH. We made a whole bunch of these during glycolysis. We made them from NAD+, but we've run out of NAD+. So we need to have pyruvate convert NADH back to NAD+. Okay, so the first one we're going to talk about is lactic fermentation. So this is what happens inside our muscle cells, okay, or when we're going to produce lactic acid. So some single-celled organisms and muscle cells temporarily lack oxygen. Both undergo lactic fermentation. Now, cells will always prefer to do aerobic. They're always going to prefer, if they can, do aerobic respiration because you maximize energy production. But if you run out of oxygen, then you're going to switch over into anaerobic and start making lactic acid. So in this case, the pyruvate created during glycolysis is used to reoxidize NADH to NAD+. When this happens, pyruvate is converted to lactic acid, or lactate is another name for it. Okay? When NADH is converted to NAD+, it allows glycolysis to continue going. Okay? So what happens to the lactic acid that's produced because pyruvate converted NADH to NAD+? Well, if you're a single cell, like a bacteria, all you're going to do is shoot that lactic acid right outside your body and then outside the environment and say, okay, well, I'm done with it. It's not my problem anymore. I've shot it outside the environment. Well, inside our bodies, we can't technically do that because if this was a muscle cell and you shot the lactic acid out, you're just going to shoot it into some other muscle cells. So we have a different way of doing it. Okay? So the lactic acid generated in our muscle cells must be reoxidized back into pyruvate to protect tissues from the acidic environment. So lactate is sent to the bloodstream. So you're going to take it out of the muscle cells that are producing it and send it into some rusting muscle cells. So if you were running a ton, you might send it to your arms in order to reoxidize lactic acid back into pyruvate, and then you've prevented that lactic acid from destroying your muscles. Okay, the amount of oxygen that's required to convert that lactic acid back to pyruvate is often known as oxygen debt, and that's just a term that when you hear people talking about metabolism, they'll use oxygen debt, and what they're referring to is how much oxygen you need to convert all that lactic acid back to pyruvate or glycogen. Okay, so we're going to do a case scenario. Just quickly, when you're just sitting there laying, okay, so if you're just sitting there watching me on the screen right now, you're doing aerobic respiration. You're not doing any anaerobic respiration. You're just doing aerobic respiration, okay? You're producing plenty of oxygen, and all your glucose is going through all the steps, producing you a ton of energy, okay? Now, you're not producing a surplus of energy, and that's key, okay? You're only producing as much energy as you need. You don't produce a whole bunch and then just store it, okay? So if you're barely moving right now, just watching the screen, you're producing just enough energy for you to be able to do that, okay? Now, if you start exercising, so say you got up and you started doing some jumping jacks, okay? As those energy requirements meet, now you're going to have to produce a little bit more energy. So you don't have this stored pack of energy, you just start making more energy when you exercise. So that's why you're going to breathe a little bit heavier. You're going to breathe a little bit heavier so you can bring more oxygen into your cells. Okay? You're also going to increase your heart rate so that all that oxygen that you bring into your bloodstream, you can move to your cells a lot quicker. So you're going to start, picking up your breathing rate and picking up your heart rate. Now, because it's moderate exercise or low exercise, you should still be doing aerobic respiration with oxygen going through all the phases because you're not bringing yourself to fatigue here. You're just moderately exercising. It's like going for a walk. Okay? You're still going to be doing aerobic respiration unless you are really, really out of shape. Right? Now, once you start picking up high levels of exercise, that's when you're now some of your cells are going to start switching. And some of your cells are going to simply be running out of oxygen. And when those specific cells, not every cell in your body, but those specific cells that run out of oxygen, so say in this picture right here, this guy's doing some squats, so right around his legs, okay, his leg cells and muscle cells in here, he's going to be running out of oxygen in his leg cells. And what's going to happen is in his leg muscle cells, they're not going to be able to do aerobic anymore. They're going to switch over to anaerobic respiration. And he's going to start producing lactic acid inside his muscle cells. Okay? And the only reason is that his muscle cells now don't have enough oxygen to produce enough ATP, so they go into their backup plan, which is anaerobic respiration. Okay, so that's lactic acid. Now we're going to quickly go over ethanol fermentation. It's the exact same process, but instead of pyruvate being converted into lactic acid, it gets converted into ethanol. Okay? Now ethanol is only a 2-carbon molecule, unlike pyruvate and unlike lactic acid. So because pyruvate is being converted into ethanol, okay, and ethanol is only 2 carbons and pyruvate's 3 carbons, it releases CO2 when we convert pyruvate into ethanol. When we convert pyruvate into lactic acid, no CO2 is created. Okay, so the popular organism that does this is yeast. Okay, yeast is going to be able to convert glucose into ethanol. Okay, so I'm just going to go over quickly basically how you make alcohol or how you make wine because it's very simple. So the first thing you need to do is to pick your sugar. So you need to have sugar because this is what we're going to start with. We're starting with glucose. So you can get this from grapes or fruit, and these would be simple sugars. Or if you're going to make beer, you're going to kind of grab a complex sugar or a complex carb. So these are like polysaccharides, and these would be like mono and disaccharides. Okay, but they're both carbohydrates. Next thing you're going to do is you're going to add yeast to it. The yeast are the organism that are going to eat the sugar. Okay, now this is the key here, is that you have to eliminate all oxygen. Because if you have oxygen there, yeast are going to perform aerobic cellular respiration because they love aerobic. So they're going to use the oxygen and then convert the sugar into CO2 and H2O. Well, we don't want that. We want ethanol. We want alcohol. So we want yeast to go into anaerobic respiration. So we have to eliminate all of the oxygen from the vat. So here is kind of a beer vat. So here's a picture of a beer vat. So they would load in a whole bunch of barleys in here and then a whole bunch of yeast. And then they have to seal it off. They have to remove all of the oxygen here so the yeast will go into anaerobic cellular respiration to make the beer. So you allow fermentation to take place. During fermentation, CO2 is produced, which also helps eliminate the oxygen. Okay, this thing would normally be a sealed container. They're just opening it up to check on it. But after they're done, they're going to close it and seal it to make sure there's no oxygen in there. Okay. The mixture is filtered and allowed to settle. If wine alcohol concentration is 10 to 15%, it cannot get any higher. The reason why it can't get any higher is because if you remember, alcohol is a poison. So as yeast make all of this ethanol, it gets a higher percentage inside this vat. So this vat's going to get 2%, then 3%, then 4%. Well, 10 to 15%, it becomes so poisonous that all the yeast die. And that's why we can't get alcohol naturally without distilling it higher than 10 to 15%. Because as soon as it gets to around 10 to 15%, it becomes so poisonous that all the yeast die. And when all the yeast die, they can't perform anaerobic cellular respiration, and they won't produce any ethanol. Okay? Now, the nice thing is that the alcohol is so high in concentration at 10 to 15% that we don't need to pasteurize it. Okay, we don't need to denature any of the enzymes of the bacteria because the alcohol has been so high that it already kills it. And that's why we don't need to pasteurize wine. Okay? Now, beer's a little different. Beer's only going to be 4 to 18%, so we cut this off a little earlier. We could make beer up to 15%, but we cut it off a little earlier because beer would taste horrible if it was 15%. But because we're cutting it off earlier, we're not killing all the microorganisms, and that's why beer needs to be pasteurized. Okay? And if it's not pasteurized, then you need to drink it pretty quickly after it's been made. Okay, so we can ferment some other products too. Okay, we only talked about the fermentation of alcohol and lactic acid, but you can ferment butanol, acetone, a number of different other processes. Okay, and then I had my kids go through the production of ethanol fuel. All right, so that's an overview of anaerobic respiration. Okay, I'm going to give you basically a worksheet that I'd like you to work on. Okay, and it's about basically. Here, I'm going to show it to you guys. So, and I'm going to give you both the worksheet as well as the answer guide, so you can take a look at both of them. But the idea here is basically it tracks the lactic acid levels of a runner. And what I want you to do is to try and figure out, well, what exactly is this runner doing? So are they sprinting? Are they pacing themselves? Are they, having the perfect amount of oxygen balance? Or do they have a surplus of oxygen, which they can start breaking down pyruvate? So here's the idea. Okay? And there's going to be a graph that hopefully pops up here. Yep, so here's the graph. So what it's asking you, so on the y-axis, I know you can't see it very well, is lactic acid levels. So this is how much lactic acid levels are in the bloodstream. Okay? And on the x-axis is time. So during A and B, so during point A to point B, they're building up a whole bunch of lactic acid. So I want you to put in here or try and guess, well, what are they doing? Are they sprinting here? Are they, recovering here? Are they pacing themselves? Then I want you to put, how you know that, and then an explanation as to why it's happening. And then from B to C, something different's happening. From C to D, something's different happening, then D to E. So I want you to kind of fill this out. It kind of just practices your understanding of cellular respiration. Okay? And then again, I'm going to have the full solutions in there as well. So take a look at it. Don't look at the solutions. Try to do it on your own, and then take a look at the solutions. Okay? Hope this helped out for anaerobic respiration. And again, a reminder that we're going to have a quiz next Thursday probably is when I'm going to put it.